Welcome everyone to the PhD exit seminar for my student, Renata Diaz. Renata started her PhD journey in my lab back in 2017 in a world that seems almost a lifetime ago now. And the pursuit of a PhD is a feat of endurance, and the milestones along the way, like qualifying exams and the dissertation defense, can be intensely stressful events for students, even under the best of circumstances. But the past 2 years have not been the best of circumstances. And Renata, like all graduate students right now, has been asked to perform this feat of endurance and stress under some of the most surreal and uncertain conditions this country has experienced in, well, a very long time. And that students are continuing to conduct research and finish their degrees under such conditions is really a continuing source of inspiration for me, and I hope for all of you who are watching your students, or you're doing it yourself, or your loved ones are doing this as they are attempting to continue to forge forward under these conditions. And this all brings me back to Renata. Over the past 5 years, despite everything going on in the world, Renata has managed to craft an exceptional and creative dissertation. The strength and the depth of her dissertation has been made possible by her truly impressive ability to cross disciplinary boundaries and in order to pursue some really deep scientific understanding. Renata is just as comfortable collecting field data from remote locations in the Arizona desert as she is in running complex analyses involving statistical physics on UF's supercomputer. This level of breadth is difficult to achieve, but doing so has allowed Renata to tackle some truly innovative and foundational questions in biodiversity research. From the moment Renata joined the lab, she never shied away from difficult challenges or complex questions. And it has been a real joy to watch her throw herself into learning the tools and skills that allow her to address the mysteries of life that she is so drawn to. So, please join me in welcoming Renata Diaz, who will take us on a tour of biodiversity dynamics that range from desert rodents to North American birds to information theory and beyond. All yours, Renata.
Great. Thank you, Morgan. I appreciate that introduction. And thank you to all of you for tuning into my exit seminar on what's a surprisingly rainy day in Gainesville. Yeah, so I'll be talking through 3 of the main projects I've completed as part of my PhD. And as is often the case in science, each of these is part of a much larger story. We're just going to be hitting the highlights. So I'll, of course, be very happy to take any questions at the end of the talk or to talk further about this work after the fact. And I'll be linking to the papers and repositories with software supporting some of this research. So with that, so the title of my dissertation is Of Rodents and Randomness, Macroecological Approaches to Community Structure. So let's go ahead and start thinking about what those words mean. So to orient you to this work, the common theme for these projects is that they derive from taking what we call a macroecological perspective to understand core questions in community ecology and, as Morgan mentioned, biodiversity science. So to unpack this, first of all, the types of questions this work deals with are pretty familiar within the usual scope of community ecology. So specifically, we're trying to understand the processes that determine how species and organisms partition resources and abundance within ecological systems, how these processes and the resulting distributions change over time, and how the stories that play out over smaller levels of organization, like population dynamics and species interactions, come together to define community level properties and distributions. The macroecological part of the equation is that we try to find a way to work with the fact that ecological systems are intrinsically highly complex within a technical sense, which is to say they're made up of lots of interacting parts that interact in numerous and nonlinear and context-dependent pathways. And that this makes it very difficult to identify general rules that we can translate across systems or even use to predict the behavior of one system at a later point in time. So in macroecology, we embrace ecological complexity and attempt to identify common recurrent phenomena that show up consistently across contexts and use those phenomena to identify potentially general processes that operate across the range of ecological diversity. And we'll come back to this later, But the key insight of a macroecological approach is to take the tools and philosophy of complex system science as it has developed more broadly in other disciplines and try to use it to understand core problems within ecology. So a macroecological approach, or at least my take on macroecological approach, is still fundamentally grounded in ecology. So the same approaches and concepts familiar to a lot of us in community ecology. So a deep interest and appreciation for natural history knowledge and the kinds of mechanistic and process-driven insights you can really only get by going and playing in the dirt and catching a bunch of rats, like the one in my hands. And in fact, all of the analyses that we're going to talk about today start out life as a case study using the data collected on these adorable little creatures. However, we're then going to be weaving in tools and approaches drawn from the wider fabric of complex system studies, especially in chapters 2 and 3. There's going to be some massive compilations of ecological data that allow us to make large cross-system comparisons. And throughout, we're going to be focusing on community-wide properties of ecological systems, so total abundance, total biomass, and the statistical distributions that describe how these quantities are arranged within communities, and looking at how those properties emerge from dynamics occurring at lower levels of organization. And to work with these kinds of phenomena, we're going to be borrowing both concepts and specific theoretical and methodological tools from other disciplines, especially combinatorics and statistical mechanics, to hopefully shed some light on ecological phenomena. So the 3 chapters that we're going to be talking about today, first we're going to be taking, throughout we're going to be taking this kind of outward moving telescopic perspective, starting from a fairly granular perspective on how empirical ecological approaches can tell us how ecosystem function is affected by species loss using a long-term experiment on desert rodents. We're going to take a step further back and use a data-intensive cross-community synthesis to examine general patterns and long-term trends in abundance of North American breeding birds, and look at how changes in community-level properties relate to changes taking place at a sub-community scale. And finally, we're going to step all the way back to where ecology starts to blur into other branches of complexity science to look at the interplay between general mathematical laws and system-specific ecological processes that combine to contribute to ecological patterns. All right, so chapter 1, about impacts of species loss on community function in the context of a changing world. I think as community ecologists practicing in the Anthropocene, we're very interested in how community function is affected by species extinctions and how this might depend on environmental conditions or in the species composition of ecological communities. For our purposes, we're going to be talking about community function in terms of either total biomass or total energy use or total metabolic flex. And so total biomass is just the sum of all of the individual body sizes of the animals in the community. And we can calculate total energy use using an allometric scaling relationship between body size and metabolic rate. And for this talk and this chapter, I'll be focusing primarily on this total energy use metric because it's a relatively direct measure of how many food resources are being processed by an assemblage and potentially made available to other trophic levels. So from the outset, we would expect that species loss would have a direct impact on community function because of the loss of the individual organisms of that species from the system. So in this scenario, in the community on the right, we have a scenario where we've lost one of the rodent species present in the community on the left. And the loss of those individuals of that species means that there's a reduction in community function in the box on the right relative to on the left. However, if the other species that remain in the community are able to exploit some or all of the resources that were made available due to species loss, we could see that these remaining species increase in abundance and partially or completely compensate for the decline in function caused by species loss. And so in this scenario, in the box on the right, the remaining rodent species has become more abundant such that both the box on the right and the box on the left have similar levels of community function, even though the community on the right is missing one of the species. And so when we see compensation, especially in a niche structured context, it's linked to what we call functional redundancy, or the degree to which the species present in a community are able to perform functionally similar roles to each other. So in this instance, exploit the same pool of resources at a particular point in time. And I emphasize at a given point in time, because functional redundancy can be dynamic over time. And there's a couple of pathways by which functional redundancy might change. So first, the species composition of a system may change, such that in this scenario, if the species were to gain the species in the bottom, it might have traits that would allow them to perform functionally redundant roles that another species might not be capable of doing. Alternatively, we could see fluctuations in the degree of functional redundancy between the same set of species due to changing conditions over time. And this is especially relevant if we were to have a system comprising species that are similar but not identical, and especially if these different species have differential responses to changing environmental conditions over time, which is a classic coexistence mechanism, as I'm sure some of you are aware. And so as a result, the consequences of species loss for community function may fluctuate over time as the degree of functional redundancy in a system changes over time. However, actually studying these kinds of phenomena can be pretty difficult because it's hard at the outset to just detect compensatory dynamics in observational data. And even if you can, it can take a long time to document shifts in functional redundancy. So this is a moment where a long-term experimental study can be really valuable. Fortunately, I work in a long-term experiment. So we're going to be looking at these dynamics using the Portal project, which is a long-term manipulative experiment on desert rodents that's set up out in southeastern Arizona. And here's some of the creatures that we get to deal with and the view we get when we're doing field work out there. So at Portal, kangaroo rats, Dipotamus genus, are really key players in the rodent community. They consume a lot of resources and are behaviorally and competitively dominant with interacting with the other rodent species. And they're also pretty photogenic, as you can see here. And at Portal, we have set up a system of fenced experimental exposure and control study plots. And so on control plots, all rodent species are allowed free access, including kangaroo rats. But on exposure plots, there's holes cut in the fencing that differentially remove kangaroo rats from the plots. And by removing kangaroo rats from the system, we can study, among numerous other things, how community function in terms of total energy use is impacted in a scenario in which kangaroo rats were to go extinct from the system. And because Portal was initiated in 1977, we've now got over 500 monthly censuses of rodent data. This is just a small subset that I was able to find in the lab last week. But this allows us to study how the effects of kangaroo rat loss for community function may change over time. And in particular, I'll draw your attention to two major changes in the rodent community that have occurred over the time series. These are regime shifts associated with the population dynamics of a particular species of mouse, which is Bailey's pocket mouse. And so in this plot, we're looking at the proportional abundance of Bailey's pocket mouse relative to the whole community over time. And the dashed lines mark the timing of regime shifts. So Bailey's was not present at the site prior to 1996, which is when it colonized the study area. And its arrival drove this dramatic reorganization of the species composition of the whole rodent community. And then there was another regime shift following a major drought in 2010. And after this regime shift, Bailey's has become quite scarce in the rodent community. So we're going to use this time series to ask how long-term shifts in community composition, specifically associated with changes in species composition and environmental fluctuations, have modulated the impacts of species loss on community function. And we're going to be looking at this by a measure that I talk about called compensation, which is the degree to which species of rodent other than kangaroo rats compensate for the loss in energy use that's caused by removing kangaroo rats from the system. And this is a quantity that's similar to a proportion in that values that are close to 1 indicate almost complete compensation, and values less than 1 indicate a lesser degree of compensation, although you can get values greater than 1 because of the nuts and bolts of how it's calculated. And here are the results of how that compensation metric has changed over time at Portal. And so the dynamic line marks the degree of compensation observed over time, and the horizontal lines and error bars mark time period wide means for time periods defined between the regime shifts that I talked about earlier. And so the major takeaway is the compensation has changed a bunch over time, and more specifically, compensation prior to the first regime shift was quite limited. Between 1996 and 2010, we saw much higher, and in fact, almost complete sometimes compensation. But that since 2010, we've seen a decline in compensation that has brought us back down to approximately the same levels we were seeing before the first regime shift. So in the 1980s and 1990s on this plot. And these changes in compensation are strongly linked with the dynamics of Bailey's pocket mouse. And the first chapter of the story has been previously documented in one of Morgan's early papers, which is to say that with Bailey's pocket mouse's arrival at the site, it triggered a pretty dramatic and sudden increase in compensation in the mid-1990s. And so Bailey's is similar in size to kangaroo rats, and the thinking was that Bailey's was able to act as a kind of functional replacement for kangaroo rats in a way that other rodents were not. And this was a clear example of changes in species composition, in that Bailey's was absent from the community and joined the community, that drove a change in the degree of functional redundancy within the community and made community function relatively robust to kangaroo rat removal. However, the decline in compensation that we've observed since 2010 has put us into a qualitatively different situation. In sort of the current configuration of the system, Bailey's is still present in the system, but it's become significantly less abundant than it was from 1996 to 2010, which means it's no longer performing the same functional role it was before 2010, but the resources are still available to it and it's still part of the rodent community. Which is to say that something about the conditions at the site or within the rodent community since 2010 have caused the degree of functional overlap between Bailey's and kangaroo rats to decrease, such that under present conditions, Bailey's isn't really a very good functional analog for kangaroo rats even anymore. And so this is an example of changes in the degree of functional overlap between the same set of species, modulating how species loss affects community function. And so the conclusions that we're going to come to in this section, there's more to the story, is first and foremost that the degree of compensation for species loss that we see within a community is highly context dependent, and that it's possible for species to compensate under some conditions and fail to under others, which can mean that as conditions change over time, it might require additional dispersal events to supply the sets of species and traits required to maintain community function, which could become increasingly difficult or even impossible, depending on how rapidly conditions change and how species are distributed geographically and how much connectivity there is between different habitat patches, all of which are expected to change during the Anthropocene. And so this work is recently accepted at Ecology. Here's GitHub repos that have links to the manuscript and the code supporting the analyses for this project. I'm also happy to take questions about it after the talk. Moving on, now we're going to take a bit of step out and take a continental scale look at how long-term trends in total biomass and total abundance have changed for breeding bird communities across North America. So as ecologists, we're pretty curious about how biological abundance has been trending over the past 30 years. Or do we have fewer organisms in the past or more or no directional change? And I personally am particularly interested in how this plays out at the community level, because that's the scale at which population level trends start to interface with species interactions and community ecology type processes like meta-community dynamics and competition to generate these system level processes. And so just as a toy example of like what I mean by like long-term trends, right? For a given community, we might be looking at a long-term decrease or an increase in abundance, or we might not really be able to pick up on much of a sort of monotonic trend at all. Although I do like to note that that doesn't necessarily mean that there hasn't been variability or change over time, just that it's not structured monotonically increasing or decreasing, such that we can capture it via a trend. And so, especially when we're working at the community level, it's important to consider how different currencies or different ways of measuring abundance might change relative to each other over time. So we can measure abundance in terms of how many like individual organisms there are in a community, or in terms of the total standing biomass of a system. And those currencies could co-vary, especially if the species composition or size composition of a system were to remain the same over time. But if there is a systematic directional shift in the average body size for a community, the long-term trend for standing biomass can become decoupled from the long-term trend for like the number of individuals. And because body size correlates so tightly with so many other functional traits, that kind of a strong size shift could be symptomatic of a significant change in the functional composition of a community. And so there's a very long and deep history of this kind of work focused on aquatic systems and tree systems. It's challenging to do this kind of work with terrestrial animal communities, generally because of two constraints. And the first of those is a lack of data on community-wide biomass. Like, we're not usually weighing all the birds in a community or all the mammals in a community that we census. And because these communities tend to have statistically pretty intractable size structures that don't play by the rules that you'll see with like a power law or something sort of similar, easy approximation. However, in the past couple decades, advances in data-driven ecology methods have made it possible to tackle these kinds of questions. And so for this chapter, I'm using data from the Breeding Bird Survey, which is a long-term continental scale citizen science project that documents the abundance and species composition of breeding bird communities in routes, 50-kilometer point counts across the United States and Canada, and using that to study the long-term trends in abundance and biomass. And so we can get to biomass data using an allometric scaling relationship between species traits and their body sizes. And recent statistical and computational methods let us get at temporal and size structure analyses for these time series. And then using the hypergator to run this kind of analysis at scale. I've been doing this with 739 routes, these analyses. And so specifically here, we're going to ask, what are the 30-year trends in abundance and biomass for breeding bird communities in North America? Do these different currencies have comparable or different trends? And how changes in the size composition of the communities contributed to decoupling between long-term trends in abundance measured in total number of individuals and abundance measured in biomass? And so here's a sort of toy example of possible outcomes in terms of those long-term trends and how the different currencies may relate to each other. So on the left, we have a scenario where change that's driven by change in the total number of individuals, which is the dark blue line, is quite similar to the change in total biomass, which is the gold line. And in this scenario, both are declining. In the middle, we have a scenario where there's been a decoupling between the two currencies. So where the change in the total number of individuals deviates from the change in total biomass. And in this instance, it's that there's a much greater decline in individuals than there is in biomass, which by definition means that the average body size community-wide has increased over time. So they're getting bigger. And that's buffered the change in biomass against the change in the number of individuals. So there's fewer individuals, but they're bigger. So biomass hasn't been impacted as much. And then on the right is a scenario where we can't really pick up on a directional trend in either currency over time. And so for 740 routes, categorize them, whether their dynamics are sort of best described by each of these three potential scenarios. And here's how those 739 routes shake out. So in about half of the routes, we're seeing some kind of a long-term trend, but the same trend to reasonable statistical approximation for abundance in terms of individuals and abundance in terms of biomass. But that for about 20% of routes, there's a significant decoupling in the trends between biomass and the trends in number of individuals. And then for another approximately a third of the routes, there isn't really a directional trend for either currency. And looking at the direction of that change, so for there's about 500 routes that have a trend in individuals, biomass, or both. And for those 500 routes, trends in terms of the number of individual organisms are dominated by declines. So in this plot, we've got a histogram of change over time summarized as the ratio of the last value to the first value. So values greater than 1 are going to be an increase, values less than 1 are a decrease. And so of these 500 routes, only about 1/3 of them are increasing, and 2/3 of the time they're decreasing in terms of the number of individuals. But then contrast this to change looking at total biomass, which is pretty much evenly divided between increases and decreases. It's pretty much bang on 50-50, 51% increases and 49% oh my gosh. The opposite of that, 51% decreases, 49% increases. It's 50-50. And so this is symptomatic of a not ubiquitous, but definitely consistent signal of increases in the community-wide body size. And so here we're looking at histograms of the log ratio of community-wide body mass comparing the end of the time series to the beginning. which is to say values greater than 0 or increases, values less than 0 or decreases. And it's broken out by whether routes are best described using a trend that's the same for biomass and total number of individuals, or the important panel is the one in the middle, which is for routes for which there's a difference in the trend for biomass versus the total number of individuals. And among those routes, so the routes for which there's a decoupling in the trend, it's almost always that the average body size is increasing over time, meaning that even though there may be fewer individuals, as time goes on, they're getting larger. And so we're getting much less of a decline in total biomass, which results in this sort of continental-wide discrepancy in the distribution of like increases versus decreases in terms of total abundance versus total biomass. And so the takeaway messages for this are that there's this, again, not ubiquitous, but consistent signal of function of size shifts that are symptomatic of potentially significant functional shifts in community composition. This increase in body size is a bit of a contrast with sort of much wider concerns about size bias defaunation in the Anthropocene, but is actually consistent with the trends that we've observed, or not we, but that scientists more broadly have observed specific to breeding birds in North America, and might reflect processes that are more specific to those systems than sort of biodiversity more broadly, and illustrates that these sort of size versus total number of organisms based currencies are non-equivalent and are telling us different things about biodiversity change. And this work, the manuscript is in prep. I would also like to draw your attention to a software package that I'm working on and hoping to release soon that makes available tools for generating these size estimates and size distributions for the breeding bird survey, which is something that I'm really excited about as a sort of second life for this kind of work, allowing folks to ask questions about size shifts in the breeding bird survey more broadly. And if that's of interest, I'd be really excited to talk about that work as well. All right, so for the third chapter, we're going to step all the way back, as I mentioned, to where ecology starts to blur into the rest of complex system science. And we're going to be using the species abundance distribution to do this, which is one of my favorite patterns in ecology. So as I've mentioned, ecological systems, ecological communities are complex systems in the technical sense, in terms of they exhibit sort of the principles of behavior of like complexity. So they have many interacting subcomponents that have stochastic, interacting, nonlinear pathways, which makes them really difficult to predict in the specific, but also makes them tend to exhibit regular emergent patterns that can provide leverage for understanding general processes that operate sort of above and beyond the kind of idiosyncrasies that occur at lower levels of organization. And these kinds of regular emergent patterns and behaviors within ecology, I think arguably the most consistent one is the species abundance distribution and its shape. So the species abundance distribution is the distribution of how the total number of individual organisms of all species in the community is divided among all of the species in that community. So how many species have an abundance of 10? How many species have an abundance of 5? How many species are present as only a single individual in a system? And no matter what specific context we're in, the species abundance distribution, or I tend to call it the SAD, is pretty dependably going to have what we call a hollow curve or a J-shaped form if we plot it this way. So with a species rank in terms of its abundance on the x-axis, and then its abundance on the y-axis. So how many species, like the least abundant species as rank 1 up to the most abundant species is in the middle plot about rank 20, and then the abundance for each species at each rank. And no matter what system you're looking at, it's pretty much going to have this J-shaped curve to it. So on the left, we have one of the communities in the breeding bird survey. In the middle is the annual plants that we have out at Portal. And on the right, we have the desert rodents out at Portal. And each one of these shows this canonical J-shaped form for the SAD. However, complex systems, for example, ecological communities, are often subject to mathematical constraints that can produce these shockingly regular emergent patterns all on their own. So there doesn't need to be necessarily additional processes in the mix to generate these kinds of patterns. And so for ecological systems, it turns out that you can generate a pretty realistically J-shaped species abundance distribution as a statistical near inevitability due to mathematical constraints on how it's possible to divide a certain number of individual organisms among a certain number of species. So here we're looking at a bunch of mathematically generated SADs that we've constructed based on total species richness and total abundance. And as you can see, they also tend to have that canonical J-shaped form that we saw in the empirical SADs. And so this might be really bad news for like complexity and ecology, because it could mean that that J-shaped species abundance distribution doesn't actually tell us very much about ecology. It's all just math, right? Maybe. But we might still be able to use the SAD to study ecological processes, because not all J-shapes are going to be the same J-shape. And there may be subtle variations between different forms of the SAD that can contain important information. So here we've added a sort of faintly dashed line, which I apologize for, marking the real species abundance distribution for the portal rodents and comparing it to a set of mathematically generated SADs that have consistent total species richness and total abundance. And just looking at this plot, you can see that the real SAD is extremely uneven. So it has a large number of very rare species, and then the most abundant species are very abundant compared to the mathematically generated SADs. And that's sort of a difference between the observed J-shape and the mathematically generated J-shapes seems to constitute an ecological signal embedded within the SAD that we can use to compare between ecological theories or to identify different processes that may be operating on SADs in different systems. So in this instance, we might ask, why are the rodents so uneven? What processes are promoting this level of high unevenness? And more generally, the way to think about this is that deviations between the empirically observed SADs and their mathematical constraints may be a path forward for leveraging the SAD to understand general processes in community ecology. Which prompts us to ask, how common are these deviations from mathematical constraints across the range of ecological systems? And what kind of clues are they giving us about the mathematical and ecological contributions to the SAD? And so to answer these kinds of questions, we have to make a couple of really important theoretical and computational advances. And the first of these is that we have to come up with a way to characterize the mathematical constraint in the SAD for a wide variety of systems. And this was made possible by a longtime collaborator, Hao Ye, who came up with a really sneaky sampling algorithm that essentially has made all of this work possible. And it's on Zenodo, there's an R package, you can play with it, it's pretty great. We also need a lot of data on the species abundance distributions for a bunch of different systems. So here I've been working with a compilation of data for more than 22,000 communities that includes trees from the Gentry transects and the forest inventory and analysis plots, a compilation of data on mammals, birds from the Breeding Bird Survey, and a collection of miscellaneous taxa, so spiders and fish and all sorts of sort of weird, less usually sampled stuff that was compiled by a student who graduated from our lab a few years ago. And finally, we need to figure out a way to quantify a deviation, like I talked about, between an empirically observed SAD and its mathematical constraint. And I won't go into all the nitty-gritty details, but as a quick run through, we can calculate summary statistics. So Simpson's evenness is the one I'm using here for a representative sample of the SADs that we can generate mathematically for a system, and then use that distribution to construct a distribution of evenness scores that we would expect just given sort of mathematical reasoning. And so that constitutes the histogram in the inset plot here is the distribution of evenness, Simpson's evenness scores for a mathematically generated SADs. And so then we can calculate that same summary statistic for the observed SAD and compare the observed evenness score to the distribution of the ones from the mathematically generated SADs. So in the inset plot, that would be the red line represents the real distribution compared to the mathematically generated ones, which are the histogram. And we can make that comparison using what's called a percentile rank, and then a low percentile rank score means that the observed SAD is extremely uneven relative to the mathematical constraint, and high percentile scores mean that it's exceptionally even. And for this example, the observed SAD actually has a percentile score of 0, which means it's exceptionally uneven relative to the distributions that we generate mathematically. And so I've done this for all of the communities, the 22,000 communities in the data set, and constructed histograms of the percentile scores for the different communities. And overall, the percentile scores are disproportionately concentrated towards very low scores, much lower than we would expect at chance, which constitutes a consistent signal of having empirical SADs being exceptionally uneven relative to those that we would expect based on the mathematical constraint, which suggests that writ large, whatever ecological processes contribute to the SAD, they're tending to encourage high unevenness that exceeds the degree of unevenness that we would expect given just mathematical forcing. And we see this to some degree across all of the data sets. However, some subsets of this compilation do behave in atypical ways. And so I'll draw your attention to the gentry transects, and these are species-rich, but relatively low abundance tropical forest systems. So out here, they have high species richness, but relatively low total abundance relative to sort of the bulk of our data sets. And they show a high signal of, they show a strong signal of high evenness, which runs the opposite to what we see in most of the other data sets. And this is something that really interests me because it could be a symptom of some sort of special processes that are specific to these kind of species-rich tropical forest systems that's promoting high diversity and low dominance. But we also can't discount the possibility that there's some additional sort of statistical edge cases that come into play when you have a large number of species and relatively few individuals. So this is an instance where there is an additional sort of tension to tease out between biological and statistical factors that generate this result for the gentry transects. And in another example, we've also found that it's much harder to detect deviations in the forest inventory and analysis plots than the other data sets. And I'll draw your attention to these plots because they tend to have relatively low species richness and total abundance, which puts them into a statistically problematic space for this kind of an approach. Because this whole sort of conceptual approach is using the logic of a complex systems science, which emerges from and operates because we can assume that we have very large number or functionally infinite number of components in a system, which generates very well-resolved emergent properties. But for these communities, for the forest inventory analysis plots, we might have 10 species and only 100 or a couple of hundred of individuals, which can sort of break down the logic of a sort of complex systems emergent properties approach. And when we have this kind of less well-resolved statistical properties, it can be much harder for us to use complex systems science to pick up on ecological signals and sort of distinguish between competing processes that contribute to the SAD. So takeaways from this work are that there are deviations from mathematical constraints present in empirical SADs, and that these seem to be able to teach us about ecological processes contributing to these distributions. But that applying this approach within ecology is going to scare up challenges that are not common in the disciplines where these kinds of approaches have sort of come up. So like in physics, you can make your assumption of an infinite number of particles. In ecology, sometimes there's really practically and functionally speaking, many fewer than infinity particles, and that has mathematical consequences. But that computational methods like the approach we've taken here, which we talk about as like a bottom-up sampling approach to generating the, or to characterizing emergent properties, can help us navigate these kinds of challenges, which I'm really excited about as a way forward for using ecology to learn things that you can't really learn from other sorts of complex systems. So this work is out in Ecology Letters, and here's links to the online repositories for the analyses and the accompanying software package that I mentioned. With that, I'd like to take a minute to just thank the folks who've inspired and guided me throughout this journey, especially my advisors and committee, and the extremely kind and patient folks in SNRE and WEC who've helped me with all the logistics and paperwork. My friends and other members of ecology over the years, and this collection of initials who I think know who they are. and of course, the entities that have supported me institutionally and through funding over the years. And with that, I would love to take any questions. I know we're a little tight in time. I also can be reached by email. And for more information, I can also direct you to my GitHub or to my personal website. And yeah, thank you so much.
Awesome. Thank you very much. That was great, Renata. And yeah, great job synthesizing so much information in such a really great way, succinct and interesting and everything. So yeah, so we do have just a couple of minutes if anybody has any follow-up questions.
Hey, Renata, I have a question.
Yeah.
I was wondering, going to the bird body size
Yeah.
data you used and the signal of a shift in average body size to bigger.
Mm-hmm.
Do you know if there's any way, or have you thought about ways to detect in which contexts that's a signal that certain larger bodied species are becoming more dominant, or potentially larger bodied species are colonizing new areas versus evolutionary changes? And then I guess another question within that is, when you're using a long-term data set like that, are you accounting for changes in body size through time, through evolution, or is it like one mean for a species?
Yeah, it's one mean for a species. So yeah, this is going to be totally, and the signal in this is driven by compositional shifts in the species composition of the system. We're working with, yeah, the size estimates are drawn from mean body size and standard deviation around that mean for, based on the CRC handbook. So it's going to be any, and it's all the records in the CRC handbook combined into one. So there's no geographic or temporal change encapsulated in that, which I think is, yeah, it makes it useful for kind of community ecology, like beta diversity stuff kind of questions, but evolutionary change or like size selective hunting or anything like that is going to be not captured in this and could be a really, really significant contributor, as I'm sure you're aware.
Thanks. Also, great talk. I really enjoyed it.
Thanks.
Okay, we have time for maybe one more question, if anybody has one, or obviously Renata had her information up there, so you can also email, but is there anybody else who has a question? Okay, well, thank you so much for this talk. This was really, really interesting. And again,